You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is November 18th, 2024. Our topic is Abbas Chapter 6, Antigen Presentation to T lymphocytes and the Functions of Major Histocompatibility Complex Molecules. This is presented by Dr. Nikita Raje. She is an Associate Professor of Internal Medicine Pediatrics and is the Chief in the section of Allergy Immunology.
I'm Nikita Raje from Children's Mercy, Kansas City, University of Missouri, Kansas City. My email's there if you have questions. And then today is November 18, 2024. Let's get started. Disclosures, nothing relevant to the talk today. Here are our learning objectives. Let's see if we can get rid of that. So the learning objectives are to talk about the antigens that are recognized by T lymphocytes. Then we'll talk about the antigen-presenting cells. We'll talk about MHC, how they process protein antigens for presentation, and then presentation of non-protein antigens. Couple of questions. We'll come back to these at the end. In the class 1 MHC pathway of antigen presentation, peptides generated in the cytosol are translocated into ER in which of the following ways? And which of the following is non-polymorphic molecule that is structurally homologous to the class 1 MHC alpha chain, associates with beta-2 microglobulin, and displays lipid antigens for recognition by NKT cells? All right, so let's get started. We'll first talk about the antigens. We have different types of antigens, and we are going to talk and concentrate on the ones that are recognized by T lymphocytes. So most T cells just recognize peptides. There are no other molecules, no carbohydrates, no lipids that are recognized by most of the T cells that we think of as our standard T cells, alpha, beta, either single positive CD4 or CD8 T cells. And the reason for this is that those peptides are the only ones that can be bound to the MHC molecules, and MHC molecules present those antigens to the T cells. So T cells only recognize those peptides bound to the MHC. They are also pretty picky and just recognize the linear peptides. If there is conformational change, so shape change, then those epitopes or determinants are not recognized. They only deal with the peptides, linear peptides that are bound to the MHC, which means they are cell associated, that expresses those MHC molecules, and the soluble antigens that are freely soluble outside of cell are not recognized by these T cells. And then the CD4 and CD8 T cells really are concentrating on each compartment. So CD4 look at the peptides that are in the cytoplasm, coming from outside to the cell into the vesicles, whereas CD8 cells concentrate on the antigens that are produced in the cytoplasm itself. So again, just kind of going through that, these cells are going to recognize peptides that are short in size and linear. They are cell associated, and CD4 binding to the extracellular antigens that are taken into the cell and packed into vesicles, versus the CD8 cells are going to recognize the cytosolic antigens, but there are a few exceptions. For example, in contact dermatitis, the peptide is pretty small, so it has to be attached to a larger hapten molecule that can be recognized by the T cells. And then some T cells will recognize non-protein antigens, but we are going to stick our discussion mainly to the T cells and the peptide antigens, and then we'll come back to the non-protein antigens at the end. Talking about antigen-presenting cells, so we know what kind of antigens are going to be, these T cells are going to be looking for, but those antigens have to be presented to them by these APCs. So what are these APCs? They present antigens to the T cells and then activate the T cells. So which ones are these? We know that there are three types of professional antigen presenting cells. These are dendritic cells, macrophages, and activated B cells. So remember, the dendritic cells are the most important, that are going to be presenting antigens to naive T cells. That's going to activate the T cells, and in the periphery, there are macrophages that can again present the antigen to those activated or effector T cells, versus and similarly there are these activated B cells that can present the antigen to effector T cells. So the most important antigen presenting cell for naive T cells, those cells are dendritic cells. So when there is exposure of microbes, when these antigen presenting cells are in the periphery or in the tissues where they are lined up and they come across these microbes, they are going to get activated, and they're going to increase the expression of molecules that will help them in their job. Their job is to present these antigens to the T cells, and in those terms it needs certain weapons, certain machinery to do their job. And so they need the class II MHC molecules, they need co-stimulators, they need cytokines, which they're going to use to activate the T cells, and they will need the chemokine receptors. So once they get their exposure to microbes, they are going to increase this machinery in the cells. Naive T cells, as I mentioned, are going to be presented with this antigens by mainly dendritic cells or other professional antigen presenting cells. But in terms of the B cells and macrophages, they are going to typically work with the effector T cells. And then, as I mentioned, apart from presenting the antigens to the T cells, they also help in activation of T cells using the co-stimulators and the cytokines. In return, there is positive feedback once there is interaction between the antigen presenting cells with the T cells. There is bidirectional interaction, which means the APCs are going to activate the T cells, but the T cells in return also provide positive feedback. So there is increased expression of class II MHC, which will make them better APCs. So that's like the bidirectional interaction. Various APCs are mentioned in this table. We've talked about the first three. There are other antigen presenting cells like various epithelial or mesenchymal cells or vascular endothelial cells that can present antigens as well. Typically, when we talk about antigen capture and antigen presentation, we are talking about the first three that we talked about. But remember, all cells, all nucleated cells have class I MHC on them that can be used to present antigens to CD8 T cells. So let's talk about the process that these cells use for antigen presentation. So the first and foremost step is antigen capture, right? For that antigen might be entering our body through various routes, skin, epithelia, or it might be produced in any tissue. These antigens are either bound antigens or cell-free antigens, right? And depending on where they are, they could be captured by different cells. For example, they could be filtered into the lymph node, or it can be free-flowing in the blood, and then they can come across antigen-presenting cells in the spleen. If they are in the tissue, they might be picked up by antigen-presenting cells at the site and then brought into the lymph node. If these are free-flowing into the lymphatic vessels and end up in the lymph node, then there might be antigen-presenting cells in the lymph node that will pick them up. So, for example, the macrophages in the marginal sinus that they'll come across, they can be picked up by that if it's free-flowing. So various ways where they will be picked up. We know the blood-borne antigens will be they will go into the spleen where they'll get filtered, and the APCs in the spleen will pick them up. How are these picked up by these antigen presenting cells? They are picked up by these processes called micropinocytosis or macropinocytosis. So small SIPs or big SIPs, or they may use receptor, which is when in that case it's called endocytosis, or it will form a kind of a pouch around it, which is called the phagocytosis. So it will use any of these processes to capture that antigen. How does it recognize these antigens? We know that these antigen presenting cells or sentinel cells will use the receptors on its surface, um, pattern recognition receptors such as TLRs, CLRs. We read about and learned about a lot of different types of pattern recognition receptors. So these antigen presenting cells will use those receptors to recognize the antigen and then use one of these methods to capture it. Once it's inside, it's going to use that and process it for presentation to the T cells. So once that antigen is captured or is bound to the receptor, it's going to activate these cells so that there are certain processes or processes that occur. For example, there will be cytokines that will be released in response to the microbes. These would be inflammatory cytokines such as TNF that will be released at the site of the capture. And that itself helps in processes that go further, that helps in transportation of the antigen from the site of infection to where they need to go. So for instance, in terms of the dendritic cells, say they are sitting in a tissue and they come across any kind of antigen that enters that tissue. It's going to capture that, it's going to recognize that antigen and then capture it. And then in that process, it will get activated. And these activated dendritic cells will lose adhesiveness for the tissue and start expressing some certain chemokine receptors such as CCR7. So they are going to be attracted to the trail of CCL19 or CCL21. So remember, the receptors are called CCRs, and then the cytokines are named CCL in this case, right? Or it can be CXCR, and so on and so forth. So in terms of the dendritic cells, when they get activated, they'll express CCR7 and will be attracted towards CCL19 and 21, which are produced in the lymph node, mainly the T cell zone of the lymph nodes. So as they start moving in the lymphatic vessel, they move towards the draining lymph nodes. In there, they're going to enter the lymph node and go towards the T cell zone. In that process of transportation, they are also continuing to evolve. So they'll express class II MHC molecules on them. They'll express co-stimulators and they'll express cytokines. So all three of these molecules are going to be used in terms of presentation of the antigen to the T cells and activation of the T cells. So by the time they reach the lymph node, they are ready to do their job. They have reached the correct site because of the chemokine receptors, but they also have the rest of the machinery, MHC class II co-stimulators and cytokines that they're going to use for their function of presentation and activation. So that's the typical scenario when we talk about dendritic cells. But as we talked about previously in the previous slide, that there are antigens that reach different regions and come across different APCs in different places. For example, soluble antigens that are not attached to a cell, they can reach the lymph node or spleen, whether it's in the lymphatic vessels or blood vessels. They're going to reach one of these places, and in there they'll be found by the APCs in those organs, and they are going to take up those antigens and process them, just like we talked about for dendritic cells. Now, one of the things that I want to point out is a process called cross-presentation. So we just talked about how these APCs are going to pick up the antigens from outside and then process them inside the cell And they likely are going to present those antigens to CD4 T cells, because CD4 T cells are meant to look at antigens that are coming from outside environment. However, there are times when there is a change in that process. So in those cases, APCs, such as dendritic cells, will ingest the infected cell and antigen that's produced in the cytosol. And instead of presenting those antigens to the CD4 cells, they'll present those antigens to CD8 cells. And that's called cross-presentation. All right, similar process of presentation occurs with macrophages or B cells, but in these cases, they are presenting those antigens to activated cells that are already coming back to the site of infection for macrophages, or activated B cells can present them in the lymph nodes which would help with their own function for processes like hypermutation of B cells and somatic hypermutation and class switching. All nucleated cells, as I mentioned, have MHC class 1 on them, and so they are antigen-presenting cells for CD8 cells. Another variation of presentation is inside thymus. The role of thymic medullary epithelial cells is to present antigens to T cells that are in development, and that helps in maturation of T cells and negative selection, so that autoreactive T cells do not move forward in maturation, and so those Tmax present antigens to these developing T cells. Also, vascular endothelial cells or epithelial and mesenchymal cells, all of them can present antigens. All right, so we talked about antigen-presenting cells. Now we move on to talking about MHC, major histocompatibility complex. So these complexes were called MHC in mice. Same genes that are called MHC have also a couple of other names, such as HLA and immune response genes. And so if you think about it, these genes were discovered because of transplantation. So when transplant tissue such as skin is exchanged between non-identical individuals, there can be rejection of those grafts. And this rejection or histocompatibility was noted to come from these genes that were on chromosome 17 of mice. So they were named major histocompatibility complex or locus, right? Now we later on there were some other experiments or other discoveries where people noticed that individuals who received multiple blood transfusions or kidney transplants had antibodies that recognized cells from the blood or kidney donors, and the proteins recognized by these antibodies were called HLA or human leukocyte antigens. Now the genes that were responsible for making these antigens or HLA, human leukocyte antigens, were found to be important for or similar to the ones that were genes that were responsible for major histocompatibility. So it was realized that it was the same genes and there were two different names given. Well, later also, again, there was another discovery where they found that in guinea pigs or mice, there was difference in the ability of antibodies that were produced against antigens, and that was inherited as a dominant trait, and those genes were named immune response genes. It turned out those genes were located in the MHC locus itself, and so overall, at the end, these three names were given to the same genes. So when we are talking about MHC or HLA or immune response genes, we are talking about the same genes. So let's talk about a process or a phenomenon called MHC restriction. So there was an experiment that was carried out by these scientists where they used lymphocytic choriomeningitis virus, LCMV, and they used strain A of this virus and injected in mice. 7 days later, they isolated CTLs from those mice and they put them in a petri dish with target cells from strain A or target cells from strain B. So different strains that were added along with the CTLs that came from the mice, which was strain A. What was noted is that if the CTLs had target cell from the same strain, they were able to lyse that target cell. But if the target cell came from another strain, they were not able to recognize that target cell and could not complete the lysis of that cell. And so the reason for this was that if the target cell has different class of MHC, it is not able to recognize antigens presented to that CTL using MHC from another strain. So talking more about the genes of the MHC locus. So MHC locus is in humans is on chromosome 6q and contains class 1 and class 2 genes that are that encode for structurally different proteins that are pretty similar, right? The locus also has some non-polymorphic genes, which means that they are gonna encode for proteins that have role in antigen presentation, but not necessarily different in different individuals. So those genes are pretty similar. The set of MHC alleles that are present on one chromosome is called MHC haplotype, which means, so haplotype is a set of alleles that are present on each chromosome. And so if someone has, say, HLA-A2, B5, DR3, like on one chromosome and has a different set of alleles on different one, we know that all individuals, even though they have two haplotypes, for some of the genes, one haplotype can be dominant, but in case of MHC genes, both haplotypes or the genes on both the haplotypes can be expressed. All right. Just showing this MHC locus, you can see some class 2 MHC, class 1 MHC genes that are located on the human HLA locus. There are some other genes that are located, such as TAPACin or TAP2, TAP1, that you might notice here, up here. Sorry if you can see these genes that are located in this region. So regardless, so we are showing these genes that can be different, class 1, class 2 MHC molecules, or the or more of the non-polymorphic genes. All right, so this figure here is showing how the MHC molecules are expressed. So we were talking about genes till now. So just kind of want to point it out to you guys. It's very confusing when I keep talking about MHC. We were talking about MHC gene locus on the chromosome till now. Now we are going to talk about the protein level, right? We are talking about the MHC molecules that come from or are produced because of those genes, and those proteins can be expressed on the cells in a constitutive manner or stimulated manner, which means constitutive means they are just there. Class 1 MHC are present on all nucleated cells, and class 2 MHC are present on the professional antigen presenting cells, B cells, macrophages, and dendritic cells. Versus the stimulated MHC molecules, which means above and beyond the constitutively expressed MHC molecules, when there are signals from TLR or when there are some cytokines that are present, there will be increased expression of MHC on these cells. What happens if there is lack of MHC? If the genes are defective, they give rise to what we know as bare lymphocyte syndrome, which means the lymphocytes do not have either class 1 or class 2 MHC. If class 2 MHC is affected, such as in a defect called C2TA, class 2 transcription activator defect, if in those cases, there are no class 2 MHC molecules on the surface of professional antigen-presenting cells. If the class 1 is defective, such as in defects in TAP2, TAP1, TAP binding protein, TAP BP, if those genes are affected, in those cases, class 1 MHC may be missing. All right, let's talk about these MHC molecules. Again, we are talking about the proteins or the MHC molecules, not the genes anymore. So these MHC molecules have an extracellular peptide binding cleft. It has a structure that we talked about last time, immunoglobulin domain, and then they of course have the rest of the structure that helps in binding to the cell, which is the transmembrane and the cytoplasmic domain. So class 1 MHC has, the table just gives you the differences between class 1 and class 2. We'll talk about class 1 MHC. It has two chains. The longer chain is called the alpha chain, and it is bound to the beta-2 microglobulin chain. The alpha chain has different domains. It has alpha 1, alpha 2, and alpha 3, three domains. Alpha 1 and alpha 2 are the ones that make the cleft in which the peptide can be located in or bound to. And then the alpha 3 domain, or the structure of that part, is pretty conservative. It doesn't change over time, and then it binds to the CD8. So remember, class 1 MHC molecule on surface of APCs is meant to present the antigen or the peptide in this A1 alpha 1 alpha 2 cleft to the CD8 T cells, and the CD8 cell has to interact with this class 1 MHC along with that peptide as well, and that interaction occurs between the alpha 3 domain with the CD8 T cell. Beta-2 microglobulin is encoded by non-MHC gene, right? It has that immunoglobulin domain as well, and it is non-covalently bound to the alpha 3 domain of this alpha chain. Talking about class 2 MHC, class 2 MHC has two chains, alpha chain and beta chain. Cleft for peptide binding is formed by alpha 1 and beta 1 domains. So it's an open cleft, and the peptides that can interact with this cleft can be a little longer, 10 to 30 amino acids, or a little longer than that in length. And the conservative part of the structure is alpha 2 and beta 2 domains. They have that immunoglobulin domain shape, and these two domains will interact with the CD4 cell. So one more fact about the genes that make this class 2 MHC. We know humans can inherit one DP and one, so the DP, DQ, and DR, those are the three class 2 MHC domains or genes, and humans inherit one DPA alpha and one DP beta gene, or one DPB and one DPA gene. Similarly, it will have the DQA and DQB genes and DRA and DRB genes, and so they are going to make these alpha chains and beta chains, whether it's A or B, attached to those DP, DQ, or DR genes. If you can kind of follow that, you know, with why there are so many genes for the class 2 MHC, because they are making both of these chains that you see in the structure of class 2 MHC, alpha chain and the beta chain. All right, here you are seeing the cleft made in class 1 MHC and class 2 MHC from the top view, and you see some of the parts of these clefts are colored red, green, blue, and the highest variability in this cleft comes in those regions that look red to you. Green has intermediate variability and blue has lowest variability. That variability helps in binding to various peptides, and that peptide cleft differs in the size of the peptide and the type of amino acids that can bind to those regions. There are 6 class 1 and 8 to 10 class 2 MHCs that can bind to several peptides, and those regions with that variability in those red regions can increase the different types of peptides that can go into that cleft depending on the size and amino acids in the peptide. Once it's bound to that MHC molecule, that peptide stays there for a little bit, so the off rate is pretty slow. But depending on whether the size or the amino acid is present in that peptide, it doesn't matter if it is a foreign peptide or a self antigen that has that peptide. As long as it has those requirements of the type of amino acids and the size of the peptide, it will be able to bind to the MHC. Right. So there are several chaperones and enzymes that facilitate the binding of peptides to MHC molecules. Once that complex is formed, they have, as I mentioned, they have slow off rate, which means they have a longer half-life, ranging from about hours to days. Right. So remember, this is something to think about. Even though the MHC molecules are able to bind to self antigens, even those, when they get presented to the T cells, it's up to the T cells whether they're going to get activated or not. So if they do get activated, if those are self-reactive T cells, they will be destroyed during development. And if they are able to bind to just the foreign antigens, they will get activated and they are able to provide that function. Right, talking about the peptide binding to the MHC molecules. So we talked about how the amino acids in that pocket or the peptide cleft is important. So these pockets are hydrophobic, and the peptides that bind to these regions have basic residue, which means there are certain types of interactions that occur between the amino acids in the peptide and the pocket of the MHC that have to occur so that there is binding of the peptide to the cleft. Right. Moving on, any questions about that? There's a lot of discussion about the type of peptides before we move on to the next subject here. So class 1, of course, has shorter peptide that can bind to it, and residues bind to the beta strands in the floor of the cleft, whereas class 2 MHC, again, there is the long peptide that extends beyond the beta strands in the floor and can interact with the alpha helical sides of the cleft. So kind of thinking through that, it's going beyond that cleft versus in the class 1 MHC. Sorry, I'm showing you. Here's the pointer. Class 1 MHC, just that in that beta portion, versus in class 2 MHC, it can go beyond that floor and can interact with these helical sides. Questions, anyone? I'm going to stop here. That's a lot of information and a lot of details. So to summarize, we already talked about antigens that can be recognized by T cells. We talked about antigen-presenting cells, And then we talked about the structure that takes part in that presentation, which is MHC. So we talked about the structure of that MHC. Now we're going to talk about how those antigens that are taken up by APCs, they are going to be processed and then loaded onto the MHC molecule for presentation. All right, so the slides that are coming up have a lot of important facts. So stop me if you have questions in between. I'm going to move past this slide and talk about protein antigen processing. Here it shows both the pathways that we are going to go into the details of. Class 1 MHC pathway on the top and class 2 MHC pathway on the bottom here. The goal is, right, we have the antigen that is taken up, whether it's from the plasmic protein or extracellular protein that is endocytosed. That's going to be processed in two different ways. Regardless of that, where and how it's processed, it's going to make those smaller peptides that can be attached to MHC molecules. The endoplasmic reticulum is where the class 1 and class 2 MHC molecule or proteins are produced. So the alpha 1 and alpha 2 chains in case of class 2 MHC, and the alpha chain along with the beta 2 microglobulin chain that we just learned about, those chains are going to come together in that endoplasmic reticulum to form the MHC molecule. Once they are formed in different ways for class 1 and class 2 pathways, those molecules of MHC are going to be loaded on with the peptides that come from the antigen processing. Once those are loaded, they are carried to the surface and they are going to be expressed on the surface of the cell. So on the surface of the cell, there is an MHC along with the peptide that is going to be presented to CD8 T cells in case of class 1 MHC and CD4 T cell in case of class 2 MHC. So let's go into the details of both these pathways. So we'll talk about class 1 MHC pathway. So we are talking about antigens that are going to be presented to CD8 T cells. So for CD8 T cells, these cells are interested in interacting with target cells that show antigens that are present in the cytoplasm of the cell. They are not coming from outside of the cell. These are antigens that are produced in the cytoplasm. So say, for example, in case of viral infection, the viral gene gets uses the machinery of the cell to produce viral proteins. Those proteins are going to be taken up by machinery called proteosomes, and they are going to be cut up into small peptides. Proteasome has special ability to cut those peptides so that these peptides are about 8 to 16 amino acids in length, and their carboxy terminal has basic or hydrophobic amino acids. These are the requirements for them to be able to be loaded onto those class 1 MHC. So the proteosomes do a good job of cutting up the peptides, but it's very organized and structured process where it cuts those peptides so that the carboxy terminal has specific types of amino acids on it. All right, so this figure also shows you cross presentation where maybe a cell that was engulfed by the antigen presenting cell or basically any tissue cell, if it engulfed that cell, say a dendritic cell engulfed a target cell, it's going to make a phagos lysosome. It's going to cut up that protein and transport that protein into the cytoplasm. And now that from the cytoplasm, that protein is going to be taken up into proteasome and cut up into peptides, as we just talked about, so that it can be loaded onto class 1 MHC to be presented to CD8 T cells, even though it came from outside of the cell. So that's your cross presentation. We're going to stop our discussion about cross presentation right now and focus on the major pathway, right, which is cytoplasmic protein, such as viral protein, that's taken up by proteasomes, cut up into peptides. And now there is a protein called TAP that helps in transporting those peptides into the endoplasmic reticulum. So TAP uses another chaperone protein called capasin. There are two types of TAP, TAP1 and TAP2, that you help in transportation of peptides from the cytoplasm into the endoplasmic reticulum. Once they enter the endoplasmic reticulum, they can be loaded onto the class 1 MHC molecules that are synthesized on the endoplasmic reticulum. So on the ribosomes or in the endoplasmic reticulum, these alpha chains are being made and being the chaperone proteins help in getting these alpha chains to interact with beta 2 microglobulin chain. Together, these two form the class 1 MHC. As this process of synthesis of class 1 MHC occurs, it's now ready to be loaded on with the peptides that are coming into the endoplasmic reticulum, transported by TAP. Once that process of loading occurs, it is transported into Golgi apparatus, and the vesicles of Golgi apparatus are gonna take or transport that vesicle to the surface of the cell, where it's going to be expressed with that peptide on the surface. That's where it's going to interact with the CD8 T cells and present that antigen to the CD8 cells. Questions?
For MHC class 1 presentation, are these cells that are actively infected by viruses that are able to go ahead and express the MHC1 complex? Is that like the most likely scenario that these cells are expressing an MHC class?
So that's a good question. So all nucleated cells are able to express whatever protein is being synthesized in the cell, and it's going to continue to sample those antigens and keep loading the class 1 MHC with those. Those can be presented to CD8 T cells, but the CD8 T cells will not get activated unless there is that second signal. Right? So second signal is important for activation, but the presentation part, the nucleated cells can continue to, remember we had talked about this with Sompara, that these are billboards, right? So the cells can continue to express and show off basically whatever is happening in the cell. And they can continue to show off whatever they are making, whatever is produced in the cell all the time, but they get the attention of CD8 T cells only when there are certain signals, when there are a lot of so when there is a viral infection or when there is an intracellular infection that causes a lot of cytokines to be produced, a lot of class 1 MHC that are expressed on the surface, that's when they gain that attention, and then CD8 T cells get that signal not only from the APC, they might also have CD4 that are activated because of that infection that are going to help with that interaction. So that attention or activation only occurs in certain cases, if that makes sense.
Yeah, that makes sense.
So all those signals, all those signals of cytokines and co-stimulators, all of those are important for activation, but the presentation it's going to continue to use the class 1 MHC billboards to show off whatever is happening in the cell.
Gotcha. Thank you.
Mm-hmm. All right. I'm going to go on and talk about class 2 MHC. Again, really important here concept. So basically a little different from class 1 MHC. So we want to know what's going on with this. Here, there is uptake of that extracellular protein, and that protein antigen is taken up into this endocytic vesicle. And now this antigen is inside the endosome or endolysosome. So when it's internalized and binds to the lysosomes have certain enzymes that can break up the protein antigens. So there is proteolysis using these enzymes in the endolysosome called cathepsins. And so when it cuts up that protein into peptides, those peptides are specifically cut in a way that those peptides will have specific properties which will help them in binding to the MHC cleft. All right, so those proteins are ready, but they are inside this endolysosome. Now, we talked about how MHC molecules are synthesized in the endoplasmic reticulum. On the ribosomes in the endoplasmic reticulum is where the alpha and beta chains for class 2 MHC are being synthesized. But wait, there can be other peptides coming inside this endoplasmic reticulum in the class 1 MHC pathway, right? The TAP and tepasin are bringing in a lot of peptides. If this cleft is not protected by something, it's going to be loaded by the peptides coming inside endoplasmic reticulum through the class 1 MHC pathway. And you don't want that. So in this case, the alpha and beta chains that are being helped by chaperone proteins to be bound to each other or interact with each other to form that class 2 MHC. There is also this invariant protein chain that helps in protecting the cleft. So it's basically blocking that cleft so that it's not taken, that place is not taken up by the peptides from class 1 MHC. Because the class 1 MHC peptides are coming from inside the cell cytoplasm, you don't want them loaded onto the class 2 MHC. So the class 2 MHC cleft is protected or blocked off by this invariant chain. Now, once it's completely synthesized, it's gonna go into a vesicle, and that vesicle is gonna interact with the endolysosome. When that interaction occurs during that process of transportation from the endoplasmic reticulum to the Golgi apparatus and inside the exocytic vesicle, that part of the invariant chain that helps with the transportation is cut up, and the rest of the part that binds to that cleft is left behind, called clip. Once it's inside the endolysosome, that clip is going to be released by interaction with something called HLA-DM. Once that HLA-DM is able to take away the clip part of the invariant chain, now the cleft is open, so the peptides that are present in endolysosome are going to be loaded onto that class 2 MHC molecule. Now the class 2 MHC molecule loaded with the peptide is going to be packaged into vesicles to go to the surface of the cell and express that on the surface and show off whatever came into the cell from outside, what it was able to capture and show off on the cell surface so that it can present that antigen or peptide to CD4 T cells. So that's the process, a little different from class 1 MHC. Here, the endoplasmic reticulum makes the molecule, but that MHC molecule is not loaded with peptides. It's transported so that it gets to the endolysosome without its cleft being loaded by peptide. Questions?
Does the molecule never go to the endoplasmic reticulum for MHC class 2?
What about the class 2 MHC?
Does the class 2 MHC never enter the endoplasmic reticulum?
It is synthesized in it. It's like the protein. So the MHC genes are going to use those genes to produce or synthesize those proteins. So it has to synthesize alpha chain and it has to synthesize the beta chain of class 2 MHC. Those two chains have to come together using chaperone proteins to make that whole class 2 MHC. So it's actually synthesized in the endoplasmic reticulum or the ribosomes, right? Where the protein synthesis usually occurs.
Okay, and then clip is just floating around in there, finds MHC.
It's not the clip, it's the invariant chain. So the invariant chain is the one that attaches to that cleft in the endoplasmic reticulum, and that helps it in transportation as well. The whole invariant chain is there till it enters, till it gets transported to the endolysosome. Once that transportation occurs, when the class 2 MHC molecule is transferred from exocytic vesicle to the endolysosome, that part of the invariant chain that was just there for transportation sticks to that vesicle, doesn't go on to the endolysosome, but the cleft is still blocked by the rest of the part, which is called clip.
Oh, I see.
So it's only a little portion of the invariant chain that stays behind, and it's still blocking the cleft when it reaches the endolysosome. So when it reaches there, the whole invariant chain is gone. I mean, the part of the invariant chain is gone, but the cleft is still blocked, and that part is called clip.
I see. Okay.
And then clip has to be taken out for that cleft to be open.
Gotcha.
Right, and that function is provided by HLA-DM. And that HLA-DM has nothing to do with the HLA genes that we just talked about. So just to be clear, this HLA-DM can be confusing because we are talking about class 2 MHC, DR, DQ, DP. Those are separate from the HLA-DM that we are talking about here. This is just showing you the class 2 MHC molecule. Really, we just talked about all of that invariant chain, HLA-DM. All right, so moving along, a few more minutes. We have a few more slides to get through. So, this is just showing that whole process that we just talked about more in detail about how those chaperones bring those chains together to form that class 2 MHC, the invariant chain that blocks the peptide binding cleft. Fuse exocytic vesicle is where it's able to transport it to that, but the invariant chain is degraded. So part of it is degraded so that there is only clip that's left behind, and HLA-DM displaces that clip. So just showing that whole process again here. Class 1 and class 2 MHC comparison, really, it's self-explanatory. We talked about the structure, how there is one alpha chain in class 1 MHC. For class 2 MHC, there is alpha and beta chain. We know the cells that express class 1 and class 2 MHC. So class 1 MHC is present on all nucleated cells. Class 2 is restricted to certain cells. We know CD4 goes with class 2 and CD8 goes with class 1 MHC. The site of antigen site of antigen processing is in proteasome in class 1 MHC and endosome and lysosome for class 2 MHC. Talked about the source of protein, antigen. This is more in detail, so looking at, what kind of subunits of proteasomes are important here, beta 1, beta 2, beta 5. Why are they important? Because they are going to make sure that the peptides that are produced, these are like the scissors, and the scissors are very specific in the proteasomes. Wherever they cut the protein, the peptides have to have certain amino acids that are hydrophobic, have to be certain length, and these subunits, beta 1, beta 2, beta 5, make sure that the protein degradation is certain type, whereas endosomal lysosomal proteases are cathepsins. Peptide loading occurs in endoplasmic reticulum for class 1 MHC, but it occurs in the endosome lysosome in the case of class 2 MHC. Important for transportation in class 1 MHC is TAP, and for class 2 MHC, we talked about the invariant chain. Cross-presentation, we already talked about this. The dendritic cells can sometimes use cross-presentation or cross-priming, which means an ingested virus or viral antigen is present in phago endosome. It's released, it comes into the cell, right? Or the target cell is taken into the dendritic cell, and then that viral protein is released in the cytoplasm. Once it's in the cytoplasm, it's picked up by proteasomes and presented to CD8 T cells by class 1 MHC. T cell response, so if it's class 1 MHC that's used, it will express the antigen or peptide presented to CD8 cells. So the function is killing off that target cell, whereas if it's class 2 MHC, it will express that antigen and present it to CD4 T cells. CD4 T cells will make Th1 response or Th2 response, or Th response or help the B cells in making antibodies, Th17 response. So it just depends on what kind of activation occurs. The response depends on that. And then again, we talked about the epitope and determinant, where those are the parts of the antigen that interact with the T cell receptor. So those epitopes that are internalized, there can be various epitopes on one antigen, and one of them might be more dominant in terms of presentation, where they are those immunodominant epitopes are the ones that are gonna end up in activating the CD4 T cells or CD8 T cells. Last in slide here, there are non-protein antigens like lipids, for example, that we'll talk about, or phosphorylated molecules that need to be presented to the T cells. And in those T cells are very specific NKT cells, which are gonna use lipids and glycolipids. They use non-classical MHC that will be used to present the non-protein antigens to these NKT cells. That non-classical MHC is called CD1. And then in case of when we were talking about the T cells, CD4 and CD8 cells, we were talking about alpha beta T cells. So the TCR is alpha beta on those cells. The TCR on 5% of the cells is gamma delta. And in those cases, there is no MHC restriction. These cells are present mostly in regions that come in contact with outside. So respiratory epithelium or genitourinary system, wherever it comes in contact with outside world. In those places, there are proteins, lipids, phosphorylated molecules, alkylamines. All of these can be presented or can be interact with these gamma delta T cells and can produce a immune response from that interaction. Okay, back to our questions. In the class 1 MHC pathway, peptides generated in the cytoplasm are translocated into endoplasmic reticulum in which of the following ways?
Should be TAP A.
A.
Sounds good. By ATP dependent transport via TAP is the correct answer. Which of the following is non-polymorphic molecule? Homologous to class 1 MHC alpha chain, associates with beta 2 microglobulin, displays lipid antigens to NKT cells.
Should be choice A, CD1.
Yeah.
CD1 is correct. Right.